Good morning, students. Welcome to this class on beta secondary kinetic isotope effect. So far we have learned important aspects of kinetic isotope effect. Let us have a recap. We had discussed the meaning of kinetic isotope effect. Kinetic isotope effect, or KIE, refers to the change in the rate of a reaction upon substitution of an atom in the reactant molecule with one of its isotopes. It is a technique which is used to determine the reaction mechanism by determining the rate limiting steps and by determining the possible geometries of transition state. In our lecture, we had also discussed the types of kinetic isotope effect. Depending upon the location with respect to the bond undergoing cleavage, KI is of two types, namely primary kinetic isotope effect and secondary kinetic isotope effect. Among these, primary KI refers to the change in the rate of a reaction when isotopically substituted bond undergoes cleavage at rate determining step. The reason behind primary KI was the difference in 0 point energies of CH and CD bonds. Due to difference in 0 point energies, the bond with lighter isotope dissociates faster at rate determining step, which leads to higher rate of reaction. And we had also discussed about the secondary kinetic isotope effect. Secondary KI is defined as the change in the rate of a reaction due to isotopic substitutions at a site other than the bond breaking site in the rate determining step of the reaction. If you remember, we had taken an example of the chloroethane. Here is the molecule of chloroethane. If it reacts with a nucleophile such as OH negative to give a nucleophilic substitution reaction, then in that reaction it is the CCl bond which undergoes cleavage during the course of reaction. And for this reaction, if KI study is done by placing a heavier isotope of hydrogen atom on the first carbon atom or the second carbon atom, then the isotopically labeled bond will not undergo cleavage and the isotope will be present even in the product. Still, if there is any change in the rate of reaction, then it will be called as secondary kinetic isotope effect, because this isotopic substitution affects the rate of reaction, but isotopically substituted bond doesn't break during the course of reaction. We had discussed the secondary kinetic isotope effect in our previous lectures. These lectures are available on YouTube, and if some student is interested, he can access them. Isotope effect. Now we know that the bond to the isotopically substituted atom doesn't undergo cleavage during the course of a reaction in secondary kinetic isotope effect. On the basis of the position of isotopic substitution, there are two types of secondary kinetic isotope effect. These are number 1, alpha secondary KI and beta secondary kinetic isotope effect. Here is a molecule, and in this molecule it is the CCl bond that undergoes cleavage during the course of reaction, and isotope can be placed on C1. This is C1. If isotope is placed here, it is called as alpha secondary Ki, and isotope can be placed on C2. If isotope is placed here on C2, it is called as beta secondary kinetic isotope effect. So, alpha secondary Ki means isotopic substitution will be on the carbon which is undergoing cleavage, and beta secondary Ki means isotope is present on the neighboring carbon. In our previous lectures, we had discussed the alpha secondary Ki in detail. Alpha secondary Ki can be defined as Ki observed when the label is placed on the carbon undergoing substitution. Again, here is the molecule where CCl bond is undergoing cleavage. If Ki is observed when isotope is placed on the first carbon, it will be alpha secondary Ki, or in other words, alpha effect occurs when the atom undergoing reaction has the associated isotope. In our previous lectures, we had discussed that observance of alpha secondary Ki can be explained on the basis of hybridization change during the formation of the transition state. We have discussed it in detail that alpha secondary Ki is observed normal when hybridization changes from sp3 to sp2, and inverse alpha secondary Ki is observed when hybridization of the reactant is sp2 and that of the transition state is sp3. This lecture is also available on YouTube, and interested students can refer to it. Now, what is meant by beta secondary Ki? In beta secondary Ki, the label is placed on the carbon beta to the site undergoing substitution. Once more, here is the molecule of chloroethane undergoing cleavage of CCl bond during a nucleophilic substitution reaction. If Ki is studied by placing a heavier isotope at this beta carbon, this is the beta carbon. If Ki is studied by placing a heavier isotope here, then the effect is known as beta secondary. So, in case of beta secondary kinetic isotope effect, a particular bond undergoes cleavage during the course of reaction, and rate of reaction is affected when an isotope is placed on the beta carbon with respect to the bond undergoing cleavage. Now the question comes, how to explain this observance of beta secondary kinetic isotope effect? To understand it, we must know that beta secondary kinetic isotope effect seems primarily when lighter isotope H or heavier isotope D is present at a carbon beta to the site undergoing a solvolysis process or cleavage, and this can be explained by the concept of hyperconjugation. To recall, hyperconjugation can be defined as an stabilizing interaction resulting from the interaction of electrons in a sigma bond, normally it is CH bond, which is present adjacent to an empty or partially filled p orbital or a pi orbital. So this stabilizing interaction is called as hyperconjugation, and it can be used to explain the observance of beta secondary Ki. In the forthcoming slide, we are going to use this concept to explain beta secondary Ki. The explanation of beta secondary Ki with the help of hyperconjugation is supported by the fact that beta secondary Ki is observed if vacant p orbital is formed in the substrate during the course of a reaction, as it happens in case when reactant converts into a carbocation. It is also supported by the fact that if hydrogen atom is present at the beta position in a geometry in which the CH bond can overlap with the developing p orbital. Now, it results into an interaction between the CH bond and developing p orbital. This interaction is known as hyperconjugation, and possibility of hyperconjugation results into beta secondary Ki. Let us understand this explanation with the help of the following representation. In the first case, it is the lighter isotope protium which is present on the beta carbon, and this is the carbon where the bond cleavage has taken place, and this carbon contains a developing or developed empty p orbital, and it has a positive charge, so it is a representation of carbocation. The presence of protium or CH bond next to the empty p orbital results into a stabilizing interaction known as hyperconjugation, and due to hyperconjugation this carbocation is stabilized. Likewise, what happens if deuterium is present in case of protium? Again, it is a deuterium or heavier isotope present next to empty p orbital of carbocation. Here again, hyperconjugation will take place and it will result into stabilizing interaction. But what is the difference between hyperconjugation due to this lighter bond and hyperconjugation due to this bond which has a heavier isotope? The answer is lighter bond, that is CH bond, breaks faster and it will create an effective hyperconjugation, whereas CD bond, which can also do hyperconjugation, but it breaks slowly, so it contributes less in the stability of developing or developed carbocation. So the beta secondary Ki can be explained on the basis of hyperconjugation stability when lighter isotope is present on the carbon beta with respect to the bond undergoing cleavage. Presence of lighter isotope leads to easy cleavage of CH bond and facilitates stability of the developing carbocation by hyperconjugation. This explanation is supported by the fact that beta secondary Ki is strongly dependent on geometrical factors like orientation of the CH group, which is going to play role in hyperconjugation. What is its orientation with respect to the developing empty p orbital? For observation of beta secondary Ki, this beta CH bond must be as parallel as possible with the p orbital. It means this is the CH group, CH bond, which is going to stabilize this carbocation by hyperconjugation. And for hyperconjugation, the 2 electrons of this CH bond will be shared with this empty p orbital. And for proper sharing, for proper overlapping, this CH bond must be as parallel as possible with this empty p orbital. If these are properly oriented, if these two are parallel, then there will be effective overlapping and there will be formation of a temporary temporary pi bond between these two C atoms, which will result into stability, and this is known as hyperconjugation. But the question is, if this CH bond is not parallel with this empty p orbital, if this CH bond is orthogonal to the empty p orbital, then there will be no overlapping. No overlapping means no hyperconjugation, no stability, and no observation of beta secondary Ki. This can be explained with the help of observation of beta secondary Ki in this structure. It is a bicyclo 222 system, and in this system, this is the CCl bond which is undergoing cleavage during a nucleophilic substitution reaction, and with respect to this Cl bond, this carbon is beta carbon, also this carbon is beta carbon. So beta secondary Ki can be studied by placing an isotope here, that is as asterisk position, or by placing an isotope on this beta carbon, which is represented by this dollar. So in actual, when beta secondary Ki study is carried out, then it was found that the value of beta secondary Ki when heavier isotope was placed at this asterisk position came out to be 1.14, and here value of beta secondary Ki was 1, or in other words, beta secondary Ki was observed when isotopic substitution was made here at asterisk position, and no beta secondary Ki was observed when isotopic substitution was made here. So what is the reason behind this? The answer is, when this CCl bond cleaves, a carbocation develops here, or empty p orbital forms here, and that empty p orbital is more or less parallel to this CH bond. So there is hyperconjugation possible here, which results into stabilization of the developing carbocation. Whereas when isotope was substituted here, this CH bond is orthogonal to this CCl bond. So when this CCl bond undergoes cleavage and an empty p orbital develops here, it cannot interact with this CH bond due to different orientations, due to orthogonalities of these two bonds. So whether it is lighter isotope or heavier isotope, it won't affect the stability of this carbocation, because at this position the CH bond cannot participate in hyperconjugation with this empty p orbital. That's why two different beta carbon atoms show two different beta secondary Ki depending on the steric factors. So dear students, this is all about beta secondary kinetic isotope effect. And finally, here are the references used to prepare this lecture. Hope you enjoyed this learning and liked it. Thank you very much.